Today, welcome to the Transplantation podcast, which is the official podcast of the journal Transplantation. I'm your host, Dr. Macey Levan. I'm an associate professor of surgery and population health at NYU Langone Health, and I'm the social media editor for the Transplantation Journal. Today, we're going to discuss an exciting and historic event in the field of transplantation, which was the inaugural Richard Slayman International Workshop on Xenotransplantation, titled Advancing Hope Through Science. The workshop brought together many of the world's leaders in xenotransplantation research to discuss where the field stands and where it's headed next. And I'm delighted to be joined by someone who has deeply shaped the advancement of kidney xenotransplantation in the world. Dr. Leonardo Riella is the medical director of kidney transplantation at the Massachusetts General Hospital and a leader in translational research focused on transplant immunology and innovation in transplantation. Dr. Riella, thank you so much for being with us today.
No, thank you, Macey, for the invitation. My pleasure to be spending some time with you.
So, this workshop carries the name Richard Slayman, whose transplant represented a major milestone in the field. Before we get into the science, can you talk a little bit about the significance of the name and why it's so important?
Yeah. Well, when we think about xenotransplantation, what really is important, I think, for everyone to understand is how important patients are for us to be able to advance the field. So there were so many important developments in the past few decades, of course, even more in the past few years, in terms of good results with gene-edited pig kidneys being transplanted into monkeys. But to move on to patients, we need patients to be able and be willing to go through a procedure and a treatment that has not been yet approved and where we cannot share any prior results for them to decide about joining or not. So through all the developments that we had in the past 2 or 3 years, I think the biggest heroes, the pioneers of all this have been the patients. And Rick Slayman, for us, it became a legend because he was the first patient in the world that was willing to receive a gene-edited pig kidney. And we went through the process with him, and we learned through the process with him of how to consent someone and what to discuss, what are the things that are important for patients, and also some of the, of course, intensive monitoring and challenges that we face after transplantation that allow us to improve and expand and finally now be part of upcoming clinical trials in xenotransplantation.
Wonderful. So, Rick was patient number one, the first living human in the world, right, to receive a pig kidney from you all at Massachusetts General Hospital. So, what strikes me about this moment is that xenotransplantation has been discussed in the scientific community for decades, but suddenly now, right, there's this new real clinical experience that is going on. So, do you feel that the field has crossed an inflection point?
Yes, I do feel we are at an inflection point. It's still early in the journey. So if we think about the journey of kidney transplantation in humans, how it started in the 1950s and kept getting better and better over time. I think in xeno we are on that same trajectory where initially there's going to be a lot of learning that's going to go on. So every patient, I think, would try to extract as much information as possible in order for us to be able to keep advancing. Small adjustments, small tweaks in order for us to keep improving the outcome. So I think it's a major inflection point. I think both for us and the physician side, for the scientists working on this, and for patients, that we're creating some hope for them in terms of an alternative to dialysis. And Rick has been, for us, this, a landmark individual because, he was the first one willing to say, yes, I want to be part of this. At the same time, what I can share is just personally is that he was afraid of it. So, even though we think about patients that some of them, went through all the process, but, as we were talking with him, he had fears, of things, not working so well, or that, things may be impacting his overall health. So, these patients, they have the courage, but at the same time, they have emotions, they have fear as well. And we had to work together with him to go through all the different steps that are required, and to ensure that, he was really willing to move forward. And most important, I think, that we've seen with many patients, and I think you, Macy, you also acknowledge the importance of the family support, the importance of others that are close to a patient who is willing to go through Xeno, to also agreeing being part of it, because truly it's one of these few trials that is not only the patient that is being recruited, but we are recruiting the patient and their team, family members or loved ones, because they all have to be part of it and work together with us to make it happen. And when we think about safety and infectious risk, there is the theoretical risk of infection transmission, and that goes not only to the patient but also people around them. So I think everyone has to be really aware of the potential risks and work closely with the team in order to move this through.
So Rick and his family were true pioneers for an entire generation of patients who will see hope from what he did.
Absolutely.
Yeah.
Absolutely.
So, for many people listening, xenotransplantation can still sound like something from a science fiction novel. But in reality, it's decades of incremental progress in genetics, immunology, and transplant science. Would you be able to walk us through some of the key breakthroughs that have allowed the field to move from experimental models to now these early clinical procedures?
Absolutely. So I would say the first big discovery that was critical was trying to identify why, when these pig kidneys were transplanted into monkeys, they rejected it immediately. They had a process called hyperacute rejection. And understanding that there was some incompatibility in blood sugars, or in the specific pig sugars. So humans have natural antibodies against certain pig sugars, just because we, in evolution, more than 80 million years ago, we lost some of the enzymes that produce these pig sugars. So we have these natural antibodies that unfortunately can cause this hyperacute rejection and thrombose an organ. So I think that was a major discovery, because once we found out what was that immunological incompatibility, we started thinking about how to address that. And of course, doing a knockout and is the first step, but more than one immunological challenge seems to be contributing to this hyperacute rejection. So part of the really game changer in the part of immunological incompatibility has been the use of the CRISPR-Cas editing tool. So these magic scissors that are capable of modifying the gene and then deleting some of these pig sugars in which we have natural antibodies against. And of course, that also permits to add some genes, for example, that are from human origin, that can help control two systems that are also very important, which is the coagulation and the complement system. So I think those two components have been crucial. And I think the third one is some of the developments that have occurred with immune suppression. So we have tested new combinations of immune suppression. I think the two critical ones that so far we have not used routinely in human transplants has been complement inhibition, so trying to control that component of the innate immunity. And now we have one of the most powerful complement inhibitors that block the cascade really on the top, the C3 inhibitor. And then we also have some drugs that block a new pathway co-stimulation called CD154. And the importance of blocking this pathway is that these drugs are capable of not only controlling the activation of T cells, but they're very powerful in reducing the activation of B cells, antibody production, and they're capable at the same time of inhibiting macrophages. So it's sort of a new immune suppressive drug that tackle some angles in which current drugs are not as effective. So, and we think that that was sort of like a secret sauce that, in combination with the genetic edits, allow us to see the incredible results that were happening in the preclinical models, in the monkey models.
Wow. So genetic engineering has been critical in overcoming barriers that really historically prevented xeno from succeeding. And now we are solving some of those immunological barriers. What are researchers still trying to answer?
So, a lot of the experiments done in monkeys, had created a sort of a landscape of what may happen in humans. But once you start transplanting in humans, we learn that we're not like monkeys, so there are differences. So I think part of it now is understanding how can we manage these patients long term. So what drugs can we reduce the dose over time? Is it the same as humans? So when we biopsy, we have all these criteria that we define rejection in a human kidney transplant. But could we just adapt and copy these criteria to the xeno, or should we adapt? And I think we're learning that we may need to adapt these criteria. So if we use exactly the same thing, it may not tell us exactly what's happening from the biological perspective. So I think there's a big learning point into it. And I think the third important point is about the infection transmission. So all of us, including the FDA, were always worried about a new infection being transmitted from the pig kidney into the recipient of that kidney. And I think we have now so good tools to monitor these patients, and at least the patients that we were involved with, we have not seen any xenopathogen transmission, which it's very reassuring. And
Yeah.
so I think this was also a major learning point in our cases. And so I think it just reassures us that, at least the infection risk is extremely low with all the monitoring. It's never zero, but I think, we can minimize that risk, to very low levels by all the testing that's done pre-transplantation in the donor. And then once they get transplanted, that all these sort of passive monitoring that happens in the blood that tries to detect an infection even before it happens. And this allows us to also have some reassurance that if something is appearing, that we're able to detect it early and potentially treat it.
Wow. I always heard zoonosis is a problem. We're concerned about this for patients, their families, and the larger community. So it is really reassuring to know that that has not been an identified issue in these early cases. Um,
That's really good.
One of the most powerful aspects of the Slayman story is that it really represents a real patient at the center of this scientific innovation. Um, what do you think is the most important lesson that we've learned so far from the earliest human experiences?
I think we got to select our patients well for those initial cases. And I think there are two aspects of that. I think number one, we have to select patients that are good transplant candidates in general. So even though in the first few cases that have happened in the heart as well as in the kidney, where the FDA was trying to push us to get more extreme patients that had very advanced diseases. In reality, we want to try to transplant patients that have the best chance of succeeding. So that criteria, patient selection, is very important, so that we don't start getting patients that were denied for a human kidney transplant, say, no, we're going to give you a xeno, because then you create so many co-founder factors that may impact ultimately the outcome. I think the second aspect is that since it is a research study, since it is a very intense monitoring protocol that is involved, I think we've learned that we have to discuss clearly with patients that it is like one scale up in terms of how often we need to see them, how often they need to get blood work, how often we may see small bumps in the road and we may have to react very quickly to get things under control. So they must be willing to work with us and be proactive as well as in reporting to us when things are not doing well or when there are any issues. So it's sort of like a partnership that is very crucial, in particular in these early stages, right? I think we want to try to minimize confounding factors, and we want to try to keep things as safe as possible for these patients. And so that very close work, the collaboration is very important. So we need that commitment from patients.
So patient selection is very important. Are you saying that these definitely should be patients that don't have access to a human donor organ or who do? I think that's been a big question in the patient community that I've heard is, well, if they're not eligible for a human transplant, why would we allow them to try this? And I think that's
Yeah.
a area of confusion. Do you have any
Yeah.
opportunity to clarify some of that for us?
I would say, and I'll give you our experience in our center. So we are very aggressive in accepting patients that may not be candidates for a kidney transplant elsewhere. What I mean by that, so if it's a patient that may have had a significant heart disease before, or may have had a cancer, that we may give them, a slightly shorter time to actually go through the transplant, or that may not have stable housing, and we actually consider them for transplantation and come up with a plan of how to follow them closely. So I think there are a lot of variables that can impact ultimately the transplant outcome. Some are completely unrelated to the kidney, the anti-rejection meds, and the potential risk of rejection. So I feel that, based on how aggressive we are, if we refusing somebody for a human kidney transplant, there are usually a very good reason for that. And most of these reasons would be things that would definitely impact the outcome on the xenotransplant. So the approach that we have taken in our center has been that we want to select patients that would be considered for a human kidney transplant to go through this protocol. If they have a living donor available, so we will encourage them to go through the living donor, get a kidney transplant. But if we look at their waiting time, and based on their blood group, their waiting time is long, more than 4 or 5 years, to get a kidney transplant. This is where we find it most useful to consider an alternative and approach them to consider xenotransplantation as an option. And of course, we cannot promise that this kidney is going to last a long time, or that he's going to benefit from it. But many of these patients, since they face a lot of challenges with dialysis, they are willing to take that step forward and give it a try. And many of them, they share, I think, the same message, that they're doing this not for them, but for all the other patients that are out there that are struggling with dialysis. And so they see this as sort of like a hope, light in the end of the tunnel that may help them advance the field and provide alternatives to dialysis, even when there's no human kidney available.
Absolutely. That's a great explanation. Thank you. I think that will go far in the patient community trying to understand some of these things. Um, I want to talk a little bit about ethics and trust and some of the global collaboration that it's taken to kind of move xenotransplantation to the clinic. And one thing that stands out to me about the Slayman workshop was that it was international and that there was international collaboration because scientific progress in xenotransplantation is happening across the world. We know it's happening in China. And bringing perspectives together seems really critical to me. So, can you talk a little bit about why global collaboration to you is important for ethics and trust around our field?
Yeah. And so I'll start by saying that, xenotransplantation is where it is right now in terms of the progress, because of all these groups that have been working around the world. They had different approaches, different organs that were studied, but they all helped, move this and with incremental knowledge for us to get where we are. And because we are in such an important stage where every patient counts and every learning that we can do is going to be so important to make the field successful, it's paramount that we all work together on this, because it's not about the success of MGH or the success of NYU or other transplant center, but it's the success of our patients that will actually determine if we made an impact or not. So if there's somebody else that's doing Xeno work and got better results than I got, I want to know what they did and I want to copy them. I don't have any problem in using that knowledge in adapting and pivoting whatever I'm doing. And so we gotta work together, and I think this is the next stage of that. I think the challenges, of course, are that we use pigs that are coming from industry, right? So we partner with industry, and of course, industry is always worried about sharing things, and publicly, because of course They also have to advance their portfolio, their drug target, which in the case of xenotransplantation is actually a whole organ. But at the same time, we physician scientists, we have to work with them and see how we can entangle and make sure that we create a network that allows sharing of information. And I think this is where I think the workshop was so important, because it was a closed door event with everyone that has done research in xenotransplantation for the past few years, that are also were involving in some of the clinical studies. And we were able to share unpublished data that was generated maybe a few weeks before the meeting in that closed environment, and try to see what are some of the signals that were present across the different studies, and what are some of the approaches that were done, and what are some of the impact of that difference in strategies. And of course, with the low numbers that we have, we cannot draw strong conclusions, but we're starting seeing things that are going to be important as we move forward. And so I think that there's this crucial, moment for us to just be trying to share things as much as possible. And I think the second thing that's very challenging with science is that it takes so long to publish something. You submit to a journal and it can take 6 months, a year and a half to get something out. And we don't have a year and a half to be sharing our experience and exchanging things. We need results, in a very fast pace in order for us to tweak or do adjustments ultimately to improve our outcomes. So it's a sort of a little bit the feeling that we had when COVID happened, where some of the research was fast paced, and we wanted to see these results right away so we can learn from each other and help things. So in xenotransplantation, we always have the same feeling that we need to share, even sometimes it's preliminary, and sometimes we don't have strong conclusions about why something is happening, but we want to share because I think it would allow us to try to learn together, put the puzzle together, and see what remaining challenges we still have facing us forward.
So collaboration and sharing and learning from all experiences is really the only way to move this forward. I couldn't agree more. I wanted to talk about another important piece to me, which is public trust, right? Transplantation has always depended on trust between patients, clinicians, and the broader public. So as xenotransplantation advances, how should the transplant community think about communicating both the promise and uncertainty of this technology as we are in this moment today?
Yeah, it's such a great question. We got to be, completely transparent. So if things are happening that are not supposed to, or there were an unexpected, whoever's participating in this trial has to know about it, so we can keep an eye on things. So that transparency is very crucial. And Macey, maybe you can share a little bit of your work as well, that some of the interviews you've been doing with patients, because that's such a important piece of the research that's ongoing.
Yeah. So, you've been a partner with me in this work to understand the patient and caregiver/care partner experience with xenotransplantation. And it's overwhelmingly positive, even in the case of graft failure, right? So we've learned from all of the patients that, like you said, I believe earlier, they're doing this because it proves a pathway for hope for people in the future. And so, even though there's been difficult times and patients have lost their graft or they've died, that doesn't mean that was, for nothing. It really has been for something. And every single case is leading towards a better outcome for the next patient. And so, the work that we're doing is to understand holistically how the patients feel about it so that we can best support them, so that we know what type of information that they need, how they need to receive that information, and how their families and communities can feel supported in their own decision-making process. So that's kind of where we are right now. It's a very small N, as you know. We've published one paper on the experience of some of the early patients prior to the start of the clinical trials. And so I think we're going to keep learning from every patient, and there's a huge opportunity to use, my favorite method of qualitative research, which is a bit different than some of the other bigger scientific methods in quantitative science, to really understand the most important questions that these patients have and what they hope to achieve. And importantly, what do they define as success?
Mhm.
What is success to a patient who's undergone this? Every patient I've spoken to is telling me that success is that we're learning something, not the number of days, not the number of hours, but success is that you, as a scientist and clinician, are learning something for the next group of patients. And I think that's truly remarkable and truly what the spirit of Rick Slayman and this whole workshop was about is supporting and honoring that patient choice and decision.
No, you couldn't have spoken better. I completely agree.
Yeah.
And the partner of Rick Slayman spoke some words in our workshop, and I think it went very deep. I think in all of us. I think anyone that was present there could feel that we need to keep pushing this forward. That there's a bigger goal that we're trying to achieve. That we will face challenges. There will be difficulties along the way, but truly having the recognition of the patients, their partners, that this is something important that we shouldn't pull back if there are challenges, and that we should keep pushing forward. I think it's so important for us and for the entire field.
Absolutely. I'm really thrilled and honored to be a part of that work. And so let's talk for a moment about the future. If we were having this conversation 5 years from now, what do you hope will have changed?
So 5 years from now, I hope that we're going to have already these clinical trials concluded, and that we're going to have a clinical approval of xenotransplantation. I think there will still be improvements. So we may have the version, 1.0, and there will be, other things that we can keep improving as we move forward. But just having that as an alternative to dialysis, it's going to be so important and game changer, and the evolution will continue. I think the beauty of, for example, having these gene editing tools is that there are no limit of gene edits that you can do, and in theory you can personalize an organ to a certain individual to match even more than what we have right now, and allow to use less anti-rejection medications. So if we look 10 15 years from now, we can start talking about personalizing an organ to a specific patient, so that we would not need to use as much anti-rejection medications, and potentially have even better outcomes than human. But I think we are on that early stage. So I think our comparison is still with dialysis. It's not with a human kidney transplant at this point.
That's very interesting to know that we're using the information now and the technology now to advance, bioengineered organs basically after xenotransplantation becomes a reality clinically. Um, so there's a lot of young researchers and clinicians that are listening and that are very interested in innovation in transplant. And I think it feels like a pretty remarkable moment for them, and a lot of them want to get involved and engaged. What advice would you give the next generation of scientists that are entering our space?
So I think in science, one of the most important aspects is curiosity, right? So that we're curious to dive deep, try to understand, have good questions, and have good mentors that would allow us to guide through, the different steps for us to be able to answer a certain question scientifically and with certainty. So, and one thing that's really important about science is that it needs persistence, it needs resilience. It's not easy. So there are a lot of paths that we take, then we hit a wall, and then we have to pivot and go in a different direction. And this is something that anyone going to science have to understand. But at the same time, there's a huge reward if you are able to move science forward. The impact becomes much greater than to a single patient. It could involve thousands of hundreds of patients in the world. So I think this is a type of reward that's very different than our day-to-day clinical work that we do, but it's so meaningful. And so I think this is the resilience, the persistence, and the curiosity, I think, would be my three greater advices in terms of how to keep advancing this forward.
I love it. Resilience, persistence, and curiosity. Those are three things that every young clinician and or scientist should be thinking about as they enter our field. So thank you so much for joining us, Dr. Riella, and for sharing your insights on what has been an amazing moment in transplantation science. And the Richard Slayman International Workshop on Xenotransplantation represents to me not just a scientific milestone, but truly a reflection of decades of collaboration, discovery, and most importantly, patient courage. As the field continues to evolve, conversations like this will help us think carefully about how innovation can move forward responsibly. And so I want to thank everyone for listening to the Transplantation Podcast. If you enjoyed this episode, subscribe and follow for more discussions on the science and policy shaping the future of transplantation. And Dr. Riella, thank you so very much for your work on organizing the Slayman meeting, as well as your work in preparing the publication, which is available now online on the Transplantation Journal.
Thank you, Mason, for the opportunity, and looking forward to seeing all of you soon.